Welcome back, everyone. Grab a seat. Today, we're doing something that feels a bit like an energetic lab meeting. We are taking a deep dive into the computational side of liquid biopsies. Now, if you work in computational oncology, you already know that analyzing cell-free DNA from a blood draw is, hands down, one of the most exciting frontiers in medicine right now. But you also know it comes with some absolutely massive data processing headaches. So, in this explainer, our goal is to compare two cutting-edge pipelines that are actively trying to solve those headaches, cfDNApipe and cfDNA uniflow. We're going to put them head-to-head and figure out exactly what each tool brings to your workbench.
Okay, let's dive into this. Here is our head-to-head roadmap for today. We'll start by looking at the cfDNA data bottleneck, check out the cfDNApipe toolkit, explore the scalable cfDNA uniflow, run a direct comparison of their features, and finally, help you choose your community standard.
Part 1. The cfDNA data bottleneck. Let's understand the hurdles in computational oncology. So you know the feeling, right? It's a shared pain point among bioinformaticians. Building custom pipelines from scratch for every single study is, well, it's incredibly tedious. It eats up so much time that really should be spent actually discovering biological insights. Why is finding cancer in a simple blood draw still so computationally frustrating? I mean, the clinical promise of liquid biopsies is incredible. Just draw some blood, sequence it, and catch cancer early. But the computational reality? It's a vast landscape of incredibly messy, noisy data. Just to ground us real quick, cell-free DNA, or cfDNA. We're talking about those short DNA fragments naturally degraded during cell turnover, just floating around in bodily fluids like plasma. They are amazing non-invasive biomarkers. But the massive challenge here is that the signals of interest, say a tiny fraction of circulating tumor DNA, are buried. They're hidden in this massive background, derived mostly from normal hemopoietic cells. Finding that tumor signal is quite literally looking for a needle in a haystack, except the hay is also made of DNA.
And this needle in a haystack problem? It's compounded by some pretty severe technical biases. We're dealing with extreme fragment length variations, simply because fragmentation is driven by natural, kind of chaotic degradation processes. Then we have GC-related sequencing biases, where sequences with high or low GC content get artificially over or underrepresented. Add in background dilution and pre-analytical handling differences, like, how long a sample sat in transit, and you've got a real mess on your hands. But most importantly, the glaring lack of unified computational standards to clean all this up, well, that sets the stage perfectly for the two solutions we're looking at today.
Now, regardless of the specific tool you end up choosing, every robust pipeline must successfully navigate three foundational steps: data preprocessing, quality control, and statistical analysis. You have to clean that raw sequence, ensure it actually meets quality thresholds, and then extract the biological meaning. Keep these three core steps in the back of your mind as we evaluate our contenders.
Moving to part 2, cfDNApipe, the comprehensive toolkit. This is a highly integrated Python package. Think of cfDNApipe as the Swiss Army knife of cfDNA analysis. Notice how smoothly the workflow flows from pre-processing right through quality control and straight into statistical analysis. But now, what's really interesting about this is its unique ability to seamlessly process not just whole genome sequencing data, but also whole genome Bissell Fight sequencing, or WGBS data. It handles the whole shebang, everything from copy number variations and single nucleotide variations, all the way down to methylation deconvolution. It really is a comprehensive, all-in-one package. Section 3, cfDNA uniflow, the scalable workflow. Think of this as an industrial assembly line built on SnakeMake.
So if cfDNApipe is our Swiss Army knife, uniflow is definitely a modular factory. Following the architecture here, the basic data processing, your trimming, merging, mapping, marking duplicates, branches out into extensive quality control using standard tools like FastQC and MozDepth. But look right in the middle, the highly recommended optional modules. This is where uniflow introduces its very sophisticated fragment length-based GC bias correction and even tumor fraction estimation via iCore-CNA. And because it's built entirely on SnakeMake, this architecture is seamlessly designed for massive throughput in cluster computing environments. Section 4, comparing pipeline features. We're going to evaluate GC bias correction and overall architecture. And this brilliantly illustrates exactly why having dedicated correction pipelines is so incredibly vital. What we are looking at here are the effects of uniflow's GC bias correction on regional coverage, specifically transcription factor binding sites for LYL1, which is associated with hematopoietic cells, and GRHL2, associated with epithelial and cancer cells. Look really closely at the blue line. That is the uncorrected signal, completely distorted by GC content biases. Now look at the orange line. That's the corrected signal. Uniflow effectively smooths out that technical distortion, pulling the signal much closer to the surrounding true coverage. Because without this correction, you might drastically overestimate or underestimate the accessibility of these tumor-relevant sites. You'd essentially be chasing biological ghosts. Let's put them head-to-head and objectively evaluate their technical capabilities. For framework, cfDNApipe relies on Python, while Uniflow uses SnakeMake. For supported data, cfDNApipe actually handles both genomic, WGS, and methylomic, WGBS, whereas Uniflow currently focuses strictly on WGS. When it comes to scalability, cfDNApipe is primarily geared toward a single computer environment, while Uniflow is fully natively cluster-ready. And for execution, cfDNApipe prides itself on simple one-command-line execution, while Uniflow utilizes a full workflow management system. So what we are really looking at here is a fundamental difference in architectural philosophy. The authors of the Uniflow paper note that while cfDNApipe is heavily praised for its highly integrated single-command execution, its single-computer design can make it a lot harder to scale across massive datasets on compute clusters. Conversely, Uniflow requires the user to adopt a full workflow management system. Yes, that's a slightly steeper learning curve, but in exchange, it offers massive scalability and deep extensibility for cluster environments. We aren't declaring a winner here, guys. Rather, we are seeing two excellent tools optimized for completely different use cases.
Section 5. Choosing your community standard. Let's get to the verdict and takeaways.
So who is cfDNApipe actually for? Well, it's the ideal choice for researchers needing a comprehensive plug-and-play toolkit. If you are working on a single powerful workstation and your research actively crosses between genomic copy number variations and epigenomic methylation data, cfDNApipe is for you. Its one-command execution is going to save you immense amounts of time and scripting headaches. On the flip side, the crucial point is that cfDNA Uniflow is your go-to optimal choice if you're operating at an industrial scale. If your lab is processing massive patient cohorts across compute clusters, if you absolutely require that deeply advanced fragment length-based GC bias correction, and you want the extensibility to plug your own custom modules into a robust workflow management system, then Uniflow is your standard. I want to end our explainer today with a slightly provocative, forward-looking question. As liquid biopsies make their massive leap from the research lab into everyday clinical practice, which standard will ultimately unify the data? Whether the field leans toward the all-in-one simplicity of cfDNApipe, the scalable industrial power of cfDNA Uniflow, or maybe even a future hybrid of both, one thing is absolutely certain. Standardizing computational oncology is the key to unlocking the true life-saving potential of liquid biopsies. Thanks so much for joining me today, and keep exploring the data.